Hello everyone. In this video we're going to continue studying elimination reactions. Let me briefly introduce to you the plan of the current lecture. We'll begin with the thorough discussion of the rate loss of E1, E2 and E1CB mechanisms. To be more specific, we'll learn the concept of the rate determining step and by using the formerly discussed energy profiles, evaluate the rate determining step for every mechanism. After that, we'll have a little pause, so we'll explore the examples of the E1 mechanism in living organisms. Finally, we'll discuss the influence of different factors such as carbocation stability, leaving group ability, the polarity of solvents, and base concentrations on the E1 mechanism. Let's go! As you can see, the names of the beta elimination mechanisms also include numbers. The same pattern you have learned while dealing with nucleophilic substitution mechanisms. But what do these numbers tell us? These numbers indicate the molecularity of the rate determining step. The rate determining step is the slowest step of a chemical reaction that determines the speed at which the overall reaction proceeds. For reactions that include more than one elementary step, the rate determining one is that with the highest activation energy. For E1 mechanism, the rate determining step is represented by dissociation of CY bond, because the corresponding activation energy is much higher than that of the second step. You have noticed that only one compound is involved in this rate determining step, meaning that this step is monomolecular. The subsequent abstraction of the proton is much faster and does not affect much the overall reaction rate. So, the reaction rate depends only on the concentration of the initial substrate and does not depend on the concentration of the base. The rate law of E1 mechanism is shown on the slide. As you can see, the rate of the reaction RE1, which proceeds via E1 mechanism, depends on the rate constant denoted as Ke1, and concentration of the substrate R containing leaving group X. The corresponding units are represented below. And what about the E2 mechanism, you would say? For E2 mechanism, the rate determining step is represented by dissociation of CX bond and simultaneous abstraction of the proton by a base. In other words, two compounds, namely base and substrate, are involved in a single reaction step. It means that the rate determining step is bimolecular. So, the reaction rate depends in this case on the concentrations of the initial substrate and on the concentration of base. The rate law of E2 mechanism you can see on the slide, of course with corresponding units. Now let us discuss the kinetics of elimination from the conjugate base. The conjugate base is the carbanionic intermediate formed in the first reaction step. Number 1 indicates the monomolecular character of the rate determining step, dissociation of CX bond in the previously formed carbanion intermediate. The abstraction of the proton during the first step is much faster than CX bond cleavage. In contrast to the E1 mechanism, it has a direct influence on the overall reaction rate because it defines the concentration of carbanion, which is involved in the rate determining step. To be more specific, the concentration of base is directly proportional to the concentration of carbanion, which is involved in the rate determining step. On the slide you can see the derivation of the kinetic equation for the E1CB mechanism. Let us evaluate it together. First, notice that proton abstraction is reversible, which means we are dealing with equilibrium. It makes it possible to formulate the equilibrium constant, which depends on the concentrations of reactants and products. After that, we can rewrite the equation with respect to carbanion concentration. Lastly, we can substitute carbanion concentration in the rate law with our new expression. It is quite complex compared to the rate laws of E1 and E2 mechanisms. In some cases, we can unite the first two multipliers to one and name it constant. We will not describe the reasons for it because it's not a subject of our today's lecture, but of physical chemistry. That is why actually both concentrations are included in rate law, despite the monomolecular character of the rate determining step. I hope it is clear for you. If not,
you are welcome to ask the questions in the comments. Summing everything up, we have learned the general mechanisms and rate laws of beta eliminations. But how do we know which of these mechanisms does the reaction follow? Well, which factors do we need to consider to correctly predict the mechanism? In order to elucidate key factors, let us have a look at the general scheme of the reaction. As you can see, there are two major reactants, substrate and base. So the first factor is obvious, base. So let us examine the second factor a bit more precisely, namely substrate. It contains the leaving group, which we have formally denoted as X. Let us add it to the list of key factors. As we have already discussed, during reactions that follow E1 and E1Cb mechanisms, corresponding intermediates are formed, carbocations and carbanions. It is obvious that substituents play a key role in the stability of these intermediates. Let us add them to the list. We have already seen in the previous chapter dedicated to nucleophilic substitution how solvation can stabilize leaving groups and intermediates. It is clear that now you have learned quite a lot of new information, so let us have a short break. During this break, we'll find an answer to the question, why E1 mechanism is so important for all of us and how the nature uses it. Let us have a look at histidine. L-histidine is a proteinogenic amino acid with unique biochemical and physiological properties. Proteinogenic means that it's used in biosynthesis of proteins. The unique chemical properties of histidine, which are mainly attributed to the imidazole ring, include proton buffering, metal ion chelation, and antioxidant activities. Moreover, histidine is nutritionally essential for human beings because it is not synthesized in our bodies. In order to get this amino acid, we need to digest histidine-containing proteins. And now you would probably ask, if we and other animals do not synthesize histidine, then which living organisms can do that? The answer is simple. Prokaryotic organisms such as well-known E. coli and plants can synthesize histidine. We are not going to study in detail the complete biosynthetic pathway as it contains 10 steps and it is quite complicated and time-consuming. On the other hand, we'll focus on the single step, namely 6th one. It is the conversion of d-erythroimidazole glycerol phosphate into imidazole acetal phosphate catalyzed by enzyme imidazole glycerol phosphate dehydratase. Phosphate is denoted as OP. From the name of enzyme, it is obvious that it is catalyzing dehydration reaction. Histidine biosynthesis is a target for herbicide and antibacterial agents, with imidazole glycerol phosphate dehydratase a key enzyme within this pathway. As a result, IGPD was the focus of inhibitor design programs with several potent herbicides in development. Some of the potent inhibitors, such as IRL 1695 and IRL 1803, are now presented on the slide. Before inspecting the mechanism, let us have a look at this beautiful enzyme. It is of octahedral symmetry and contains 24 subunits and more than 33,000 of atoms. You can see the mechanism of dehydration process on the screen. During the first step, the resonance-stabilized carbocation is formed and OH leaves as hydroxide anion. It is quite an unusual situation when OH group acts as leaving group in E1 mechanism. The formerly discussed enzyme traps hydroxide anion with acidic proton colored red, thus leading to formation of water molecule. Then proton in beta position is abstracted and new pi bond is formed. Finally, the product is tautomerized to imidazole acetal phosphate. To my mind, this reaction serves as a perfect example of E1 mechanism in biology, and it could be important for agriculture and pharmaceutical sciences. Now, it is time to finish the break and return to our main topic of the discussion. Now, I would like to go to the next part, where we will be discussing factors that favor the monomolecular substitution mechanism E1. Look at this graph. The reaction here proceeds via an intermediate carbocation. So, the more stable carbocation is, the better it is for the E1 mechanism. We have already learned in the previous chapter how the stability of carbocation depends on its structure. The least stable is methyl carbocation followed by primary carbocations. These two groups are so unstable that do not literally form in the solution. According to the scheme presented on the slide, secondary carbocations are more stable than primary, but significantly less stable than tertiary. It means that secondary alkyl halides also literally never follow E1 elimination mechanism. In contrast, tertiary halides are the best substrates for the E1 mechanism. Until this moment we have discussed only alkyl carbocations. However, there are other types of carbocations. For example, pi conjugation is quite an effective way of stabilizing carbocations. Such conjugation enables to formulate more resonance structures, for example, for allylic and benzylic carbocations. That's why allylic carbocations are less stable than tertiary alkyl carbocations, but more than secondary. And benzylic is the most stable among the discussed carbocations. It means allylic and benzylic derivatives can follow E1 elimination mechanism. In order to form the pi bond, both benzylic and allylic carbocations should be substituted. For example, benzylic alcohols follow E1 elimination mechanism under strong acidic conditions, like in the presence of sulfuric acid, and yield styrene or its derivatives. The dehydrohalogenation of dimethyl substituted allylic bromide and methanol, which results in the formation of diene, represents the E1 mechanism for elimination from allylic derivatives. Another way of carbocation stabilization is heteroatom in alpha position, which is capable of free electron pair donation. It also makes it possible to formulate more than one resonance structure, thus stabilizing the whole system. You can see examples of such substituents on the slide, such as chlorine, bromine, O-methyl, NME2, etc. That is why we can add such carbocation to the list and put a tick nearby. The effect of the leaving group is obvious. The better it is, the faster carbocation is formed, thus favoring E1 mechanism. We have already learned the comparison scheme of various leaving groups in the previous chapter, so we will not put much emphasis on it now. If you want to refresh your knowledge, you can briefly examine the scheme presented on the slide. We have already said that solvent can also have a significant influence on the mechanism according to which the elimination will proceed. And again, as we know, protic polar solvents favor SN1 reaction because they can solvate the leaving anion properly, thus decreasing its energy. The same trend can be observed in the case of E1. Polar protic solvents such as alcohols, carboxylic acids, and water-based mixtures stabilize the leaving group and the forming positively charged intermediate, carbocation, because they represent good ionizing, ion-solvating media. That's why they favor E1 mechanism. There are lots of polar protic solvents, some of them can be seen on the screen. The most acidic protons are colored red. The numbers below the structures are corresponding dielectric constants. The higher the number is, the more polar is the solvent. The last factor to discuss is base. Being more specific, its concentration and pKb value. If one examines the rate law of E1, he will find out that base concentration is not a part of the equation. The concentration of base does not affect the rate of E1 reaction, so it cannot favor or disfavor it from first sight. Further investigation of competitive E2 mechanism, however, will show completely other dependence. The higher is the concentration of base, the higher is E2 reaction rate. That's why high base concentrations favor E2 mechanism and thus indirectly disfavor E1. Let us evaluate the influence of base strength. Look at the reaction presented on the slide. Alkene is protonated with BH+, yielding carbocation and conjugate base B. It is quite complicated to protonate alkene. In order to do it, you need to use some strong acids such as hydrobromic acid. Do you remember Bronsted acid-base theory from your inorganic chemistry course? One of the key postulates of this theory is that strong acid has a weak conjugate base and vice versa. On the slide, the weak base is alkene, so the conjugate acid, carbocation, is a strong acid. Consequently, carbocation formed during E1 mechanism will give its proton to any particle, which can act as a base. In the solution, the probability of collision with solvent is much higher than of collision with any other base, so carbocation in E1 usually reacts with solvent. That is why the strength of the added base is not important and does not favor the E1 mechanism. In the next video, we are going to discuss the competition between SN1 and E1, so you will better understand why this mechanism is not well suited for synthetic applications. And here again is a brief summary of factors favoring E1 mechanism. Stabilized carbocation, good leaving groups, polar protic solvents, and small base concentrations. I hope this part is clear for you. If not, you know where the comments are.
That's it. Let's make a small recap of what we have learned. We have begun with a thorough discussion of the rate laws of E1, E2, and E1CB mechanisms and found out the corresponding rate determining steps using the formerly discussed energy profiles. Then we had a little pause and discussed the role of the E1 elimination mechanism in biochemistry. The last topic was quite large. We evaluated the influence of different factors such as carbocation stability, leaving group ability, the polarity of solvents, and base concentrations on the E1 mechanism. In the next video, we'll evaluate the influence of all discussed factors on E2 and E1CB mechanisms. Moreover, we'll have a little pause and learn about the reaction that proceeds according to the E2 mechanism in the citric acid cycle. If you have any questions related to this video or any topic in chemistry, biochemistry, or pharmaceutical sciences, you are more than welcome to ask them on our website or in the comments below. Don't forget to give us a like and subscribe to our channel. Thank you very much for your attention, and we'll meet once again in the next video.